Okay, so now we're going to talk about secondary immunodeficiency. And one of the big things about secondary immunodeficiency, this means that this is not part of the cell. This is coming from something exterior from the cell and causing immunodeficiency. And of course, when people think about this, they think about HIV, which is going to be a major target of this. And so you can think of secondary immunodeficiency as being related to all of these substances. Infection with HIV, age can have an impact, whether you are very young in developing um, baby or an older person, elderly person. Also, drugs, cancer, whether you're going through chemotherapy and immunosuppressive drugs, nutrition, etc., can all have an impact on a secondary immunodeficiency. And if you do have a secondary immunodeficiency, you might be susceptible to infections. And these infections can then be our three: recurrent, unusual, and difficult to treat. And so we can also be susceptible to tumors. And so with respect to HIV, there's approximately 40 million people infected worldwide. About 2 million infected every year. And about 1.2 million die every year. Now, this, of course, HIV is a chronic infection. So everybody who's infected every year or who is infected are not going to die during that year. This is very different than an acute type of infection. But it is still a high burden on the world population. And so we're going to take some time to talk about HIV with respect to immunodeficiency. So HIV is an RNA virus similar to SARS-CoV-2 and is considered a positive RNA virus. And so a positive RNA is equal to what we consider messenger RNA. So the positive strand RNA can make proteins. That's what that means. It is a very simple, it has 3 genes, which through alternative splicing makes 9 proteins. HIV is a retrovirus, and this means it can go RNA to DNA. And then it has 2 glycoproteins. You have GP120 and GP41. And 120 is going to bind CD4, so it infects CD4 cells. And GP120 also binds CCR5, which is a chemokine on CD4 cells. And so both 1 and 2 must take place, and then GP41, 3, can fuse with the cell membrane. And then you get infection. Now, what this means is HIV can infect CD4, macrophages, DCs, and of course monocytes. And it's really within the macrophages that it sets up a latent infection. Now, this does not mean necessarily it's a latent virus, so I want to make this very clear. HIV replicates. It's going to replicate inside of cells. However, it can hang out and hide inside of other cells. So one of the problems with HIV is that you have to eliminate every cell that's infected. And so when this RNA goes to DNA, this is going to incorporate into the host genome. And so it becomes part of that cell. And so when that cell divides, HIV divides. When that cell divides, HIV is in the parent cell and the daughter cell. So HIV becomes part of your genome. So again, HIV is going to be a virus that has an envelope, GP120 and GP41. It's made up of RNA on the interior. The GP120 is going to bind to CD4 on our CD4 T cells or our macrophage or dendritic cells, and then they're going to bind to a chemokine receptor in our CD4 T cells. This is going to be called CCR5. You then get binding of CD41, and CD41 is going to expose an area that's able to incorporate into the membrane. This is called the unzipping. And then it's going to zip close and fuse with the membrane and release the genome into the cell that it's trying to infect. Now, HIV has reverse transcriptase. Which is called RT. And this is basically going to allow it to go from HIV plus RNA to HIV double-stranded DNA. And then this can get into the genome. Now, with the reverse transcriptase, you have errors, and there is no correction. And so 1 in 3 HIV life cycles is a new mutation. So within a week of infection, a person has many mutations, mutated viruses. And this is typical of RNA viruses. And then once inserted into the host genome, it becomes similar to host genes. Which means that it's silent until activated. Now, in macrophages, it can become latent because a macrophage typically isn't activated. It's kind of doing its homeostasis thing, but when a macrophage becomes activated by a bacteria or a fungi, then the virus can get activated. But macrophage latency is a reservoir for the virus. It doesn't mean it's not circulating somewhere else. It doesn't mean it's not proliferating somewhere else, but within a macrophage, it can kind of hang out until that macrophage gets activated. Now, the virus is cytopathic, which means it'll kill the cell it's infecting. Now, not all viruses are cytopathic. We talked about hepatitis B, for example, is not cytopathic. It's the immune response that causes the toxicity. With respect to HIV, it does kill the cells. And so, for example, here we have our HIV particle that fused with our membrane. It released the genome into the cytoplasm. It has reverse transcriptase associated with the virus particle, and that's going to start to produce a copy of the DNA, which is this red region here. And then the copy of the DNA gets inserted into the host genome. And then when the host gets activated by some way, and it gets those transcription factors that we've talked about with respect to activation and FAT and NF-kappa B, then you get transcription of the HIV genome. Now, some of the HIV RNA again, is going to be plus RNA, which equals messenger RNA, which can become proteins. And so that's what happens here, it becomes proteins. And then some of the RNA can become genome, because this is a plus RNA strand. So the genome gets packaged into the virus particle and then gets released. Now, some of the immune responses to HIV is that we have, for example, a plasmacytoid dendritic cell, which is a good type 1 interferon inducer, which we know is antiviral. However, a plasmacytoid dendritic cell function is impaired following HIV. And HIV replicates to very high titers. And so we're going to have high levels of antibodies, which is diagnostic. So you can measure antibodies from GP41 and GP120. Now these antibodies are not going to impair the binding, so they're not neutralizing, because they're binding to epitopes that are not important in the virus binding to the receptor. The binding sites for the CD4 and chemokine receptors are actually inaccessible to the antibodies, so it's almost like they're hidden. So HIV hides important epitopes from the antibodies. Antibodies might be made to these epitopes, but they can't get access to the actual antigen. Now, the virus is also going to cause high mutations because of the reverse transcriptase. And this is going to occur for every infected cell. And so you have to have primary CTL, cytotoxic or cytotoxic lymphocytes, plus B cell activation constantly. So it's like you get one set activated and then the GP120, GP41 mutate, then you have another T cell, B cell response, and then they mutate, etc. In addition, your cell-mediated immune responses can target and eliminate active replicating cells, but CTL cannot target latent cells. And so, again, it goes back to you have to kill every single cell that's infected with HIV that has integrated virus into the genome. And if you can't do that, then your HIV is going to be able to survive. It can wait until it can get reactivated, and then the immune system has to change and shift in order to respond to the new emerging strains that HIV produces. Okay, so with respect to HIV, keep in mind that the virus can kill cells. And keep in mind it's going to be CD4, macrophages, DC, so this is going to lead to immune suppression. And so this is cytopathic effects. Which is CPE. Your CD4 cells are going to be killed by CD8s. And that's through MHC class 1. You have widespread activation, which is going to lead to activation-induced cell death. Again, it's that T cell that thinks that it potentially can be autoreactive because it keeps seeing the antigen over and over again, but it's because it's chronic disease. Now you're going to have the immune system, so you're going to have some clinical. Let's talk about clinical. So the first is your immune system will control the virus. And then you're going to have a HIV seroconversion illness. And this is about when antibodies develop. And remember, antibodies kind of come up after T-cell mediated. And this coincides with a rash, malaise, or fever. And then you go through an asymptomatic stage. And the virus is still replicating. So you have low-level virus replication plus CD4 death equals CD4 replacement. So you're not at that critical stage yet. And then you're going to have a stage where your CD4 count falls. And this is going to affect Th1 plus Th17 opportunistic infections. And so this goes to this chart here. So when your CD4 count is at the normal level, you don't have diseases. But as it starts to fall, so now you go from 400 to 300, you start to get reactivation of those infections that can be reactivation with minimal immunodeficiency, stress, and things like that. So that would be herpes simplex, herpes zoster, shingles, things like that. When you start to drop again, you get an increase of tuberculosis and candida. These are going to be Th17 and Th1. So those are both infected because your CD4 T cell counts are going down. And then you're going to have another drop and you're going to get in more opportunistic type infections. Pneumocystis jirovecii, for example. This is just common in the environment, and most of us don't have any problems with jirovecii. But if you are immune compromised, you start to present with those diseases. Toxoplasmosis, this is the cat litter disease. And again, if you have a cat and you've been exposed to toxoplasmosis, you've been infected with it, it's a chronic infection, you don't get rid of it, but your T cells keep it in check. As you lose the ability of your T cells to function, you lose your CD4 T cells, then you're not able to keep the toxoplasmosis in check and you got a disease now. You have herpes, human herpes virus 8, which causes Kaposi sarcoma. Again, another herpes virus that usually does not cause any problems in a normal, healthy individual. And now you start to have cancer associated with that. You get a non-Hodgkin lymphoma. Comes by EBV transformation. Again, EBV causes mononucleosis, creates a chronic infection. You're able to keep it in check again until you become immune compromised. And then as you got dropped even lower levels, you start to get normal, common, everyday pathogens or non-pathogens causing disease. So you have your mycobacteria that never caused disease in healthy individuals all of a sudden causing a problem. You have an outburst of cytomegalovirus infection that, again, your immune system was able to keep in check, but no longer can. Your invasive fungi, which are normal environmental potential pathogens, but your immune system can handle unless it becomes compromised. And eventually you will succumb to these opportunistic infections. Now, with respect to HIV outcomes, one thing to keep in mind is there is no cure. It is chronic manageable illness. That is almost 100% preventable. This is not a virus that spreads similar to SARS. It's a very hard virus to get, and if you take certain precautions, you should be able to prevent it. However, those precautions are not always taken, and so we have the pandemic that has been with us for now going on about 3 decades. So some of the outcomes. So with respect to outcomes, you have polymorphisms in CCR5. That's that chemokine receptor. So remember, HIV has to bind to CD4, then it has to bind to a chemokine CCR5. So if you have a polymorphism in CCR5, typically this leads to a decrease in HIV ability to bind. So the homozygous, therefore, this is my therefore, have a decreased risk. You also have cytotoxic T cells. Responses are going to vary. And this is going to be caused by HLA. So if you have heterozygote, this leads to an increase in better outcomes. And it's just what we've been talking about. If you have diversity within your HLA repertoire, you're going to have diversity in the ability to present antigens to your immune response, and your immune response is going to adapt better to the changing HIV antigens. Now, another thing is that you have treatments. We have heart, which is highly active antiretroviral therapy. And so for highly active antiretroviral therapy, it's going to be a combined therapy. It was very clear that very early on, a monotherapy was not working because the virus would mutate too quickly. So the idea was to put together a combination of targets with respect to our antiviral drugs. And so the likelihood of one virus having a mutation that would be protective or would be escaping all of the different drugs would be very low. You can have one mutation that will confer protection against a particular drug, but the other drugs would be able to eliminate the virus. And so the drugs are going to typically be a reverse transcriptase inhibitor. And this is typically a nucleoside analog. And a nucleoside analog is basically going to pretend that it's like an A, a T, a G, and a C. But for example, the simplest one will have at the 2' an OH or a death sequence so that you cannot add any more bases to it. So that's a nucleoside analog. And then you're also going to have an integrase inhibitor. Oops. And so this is going to prevent the incorporation of the double-stranded DNA into the host genome. And then you're going to have a protease inhibitor. And this, of course, is going to not allow any structural proteins to be made. And then you're going to have a fusion inhibitor. And this is going to block GP41 fusion functions. And now with respect to a vaccine, there is no vaccine available. And one of the biggest problems is mutations. And so many vaccines have been tested and tried, targeting B cells, targeting T cells, targeting both B and T cells. But the virus mutates so much that even if you get a protective immune response, there is another mutation in the virus that will expand, that will be able to escape the vaccine-induced immune response. And then your body has to respond to that virus again. So no virus is available yet for HIV. Now let's talk a little bit about other factors that can influence immunodeficiency. One of the things is age. So you have infants, and they're basically immature. Everything is a primary response. And so you have fetal antibodies that are going to be made starting at about 20 weeks. And this is of gestation. And then you have adult levels of IgG are going to be present in approximately 5 years of age. So it takes time for the child, the baby, to make enough antibodies similar to a level that you see in adults. And we have maternal antibodies, and they're for about 6 months. They'll protect the fetus. You also have IgA in the breast milk, which will protect the fetus. And this is going to protect mostly the lung plus GI infections. Now what's interesting is a fetus that is bottle fed has about 60 times more likelihood to get pneumonia. Now, of course, premature is most at risk, and they're going to, this primarily caused to the decrease of maternal antibodies that are transferred to the fetus during those last few weeks. And then you also have an immaturity of lung surfactant. That is also going to play a role in increasing of respiratory illness. Now on the other side, you have aging. And aging, you're going to suffer more infections. This is considered when you have mild immunodeficiency. Your T cells memory is lost more than B cells. And then your thymus shrinks approximately 3% per year. through the middle age, and so you have less thymocytes being produced, and proliferation of your thymocytes in the periphery are what are going to make up most of your T cell repertoire. You also have CMV infection, and this is going to take up most of your T cell response. So you're going to have poor responses to vaccines. Of new pathogens. So I just want to use really quickly measles as an example for this, especially with the young. Okay, so we have a difference in the measles vaccination in developed versus underdeveloped world. So we have here our magnitude, and over here we're going to have, oops, again, of time. And here we'll do 6 months after birth and then 1 year. So in the developed world, the risk of measles is low. And so we're going to have maternal antibodies that are going to wane. And then what we're going to have is we're going to have a threshold of protection from measles. And then we're going to have another threshold, which is clearance well, actually, I'll call it prevention, maybe that'd be easier. Prevention of vaccine. So basically what this means is that the amount of antibodies, maternal antibodies, to prevent infection of measles of a child, of a baby, is higher than the amount of maternal antibodies needed to neutralize the vaccine. And so what this means in the developed country where you have low risk to measles is that you have the vaccine is given at year 1. Now when you take this image, and we're going to look at it from the perspective of a developing country, now what's changing is the risk is high. And so they can't really wait until a year of age to give a vaccine because the risk is too high. So the vaccine is given at about 6 months or 9 months. So sometime between 6 and 9 months is where you give the vaccine, even knowing that there might be inactivation of the vaccine by maternal antibodies because the risk is just too high to leave the infants for 6 months susceptible to measles. And so they use the vaccine to bridge the gap for where you're protected by maternal antibodies and then left susceptible because of the lack of maternal antibodies. Okay, so let's talk just very briefly about additional impacts. So we have drugs. Drugs are going to be very common. There's a lot of different drugs that can be given that can cause immune deficiencies. And what's key about drugs is that removal of the drug removes the deficiency. And so some drugs are going to be treatments for cancer. And these can lead to neutropenia. And then, of course, you have problems with extracellular pathogens or corticosteroids. Are going to damage B and T cells. And then you can have also anticonvulsants, which can impact B cells. You also have cytotoxic drugs that are going to be used for autoimmune and transplant prophylaxis. And these can eliminate T and B cells, for example. B cell malignancies are where you have a large amount of monoclonal antibodies made. And these are referred to as paraproteins. And therefore, they're going to lower the ability to respond to pathogens. You also have thiomas. Oops. Again. Which is rare and can lead to you also have kidney and gut diseases. These are where you have protein loss, and one of the major proteins that can be lost are immunoglobulins. So if you have renal damage plus protein loss equals Ig loss. So whatever is circulating, you can lose. You can also have severe diarrheal disease, which again loses immunoglobulins. Nutrition, zinc and magnesium are important for cell-mediated immune responses. And stress. So you can have epinephrine, corticosteroids, which lymphocytes have receptors for. And this is going to be immediate, and this is going to be more chronic. And then infections, you have, of course, our CMV in elderly. You also have, with respect to CMV, the T cell response is going to become oligoclonal, so too many cells with CMV specificity. And so you don't simply have enough to respond to other pathogens. And this is primarily in elderly. And then you also have malaria and rubella, which are going to have antibody deficiencies. And then measles infects CD4 cells, so you get a transient immunodeficiency. And most people die from pneumonia. And it could be months after measles. So measles, like your HIV, infects CD4 T cells, but measles is not a chronic infection, does not incorporate into the genome, and so forth. It's therefore considered transient, but it can last up to 6 months. So it's a pretty nasty disease. Okay, so that is the end of our secondary immunodeficiencies.